class we have completed few industrial products and we will be continuing the industrial products. In this class we are going to discuss the role of microbes in industrial products, especially in chemicals, enzymes and bioactive molecules. See, microbes have been used in the commercial and industrial production of certain chemicals. Certain chemicals like organic acids, alcohols, enzymes, vitamins and bioactive molecules. You may be heard about the vitamins, enzymes, etc. that you have studied in the biomolecules chapter. And organic acids are nothing but the acids which are present in an organism. They are called as organic acids. Then what are these bioactive molecules? Bioactive molecules are those molecules which are active or functional only in the living systems or can interacting with the living components. When we discuss these biomolecules, you will come to know that these biomolecules are functional only in living systems or can interacting with the living components, and a number of chemicals will be obtained from the microorganisms. We will discuss one by one. First, we will discuss organic acids. Organic acids. As I said, organic acids, these are the acids which are obtained or which are present in an organism. They may be present in a plant or they may be present in a microorganism, etc. If they are present in the organism, they are called as organic acids, and a number of organic acids are manufactured with the help of microorganisms. And in the year 1922, a scientist named as Miller. In the year 1922, Miller reported for the first time. For the first time, he reported the accumulation of citric acid. He reported the accumulation of citric acid in the fungal culture, which means in some fungal culture, some fungi will be releasing or producing citric acid. Then which is that microbe or fungi which produces citric acid? The main microbe involved in the production of citric acid, it is called Aspergillus niger. Aspergillus niger. The name of the microorganisms are very important. So citric acid, it is one of the important acid which was first reported by Miller in the fungal cultures of Aspergillus niger. Similarly, acetic acid. So this is also one of the acetic acid. It is also one of the organic acid, that is acetic acid, which is obtained from the bacteria Acetobacter aceti. And later, butyric acid obtained from the bacteria Clostridium butylicum. And lactic acid, lactic acid is obtained from Lactobacillus. So these are several organic acids which are mainly obtained from these microorganisms, and all these organic acids now they are produced in fermenters. They are also called as bioreactors in large scale and are used for various purposes. All these organic acids, they are produced in a large quantity in fermenters or bioreactors and used for various purposes. First we will see how this citric acid is obtained. So this is citric acid. It is obtained from the fermentation with the help of a microbe called as Aspergillus niger. So this is one of the fungal species Aspergillus niger, and not only Aspergillus niger, few more fungi also produces citric acid. They are namely Mucor and Candida lipolytica. So these fungi also produce citric acid, but the major one, the main microbe involved in the production of citric acid, it is Aspergillus niger. Then this acetic acid is obtained from Acetobacter aceti. Do you remember during vinegar production I told you the ethyl alcohol gets converted into acetic acid, which means during fermentation of alcohol with acetic acid bacteria called as Acetobacter aceti, which produces acetic acid. So Acetobacter aceti produces acetic acid. Then this Clostridium butylicum produces butyric acid. The butyric acid is mainly obtained from the fermentation of Clostridium butylicum. And lactic acid is obtained from lactobacillus. This lactic acid, it is the first organic acid to be produced from the species of lactobacillus and also streptococcus. Some streptococcus species also produces lactic acid, but the major one involved in the production of lactic acid, it is lactobacillus. So these are the organic acids. See, in your study material, we have given the table in which there are list of organic acids and from which microbe the organic acid is obtained and what are the uses of the organic acid. All we have mentioned in that table. You can refer to the study material. So that is about organic acids. And alcohols, you know that Saccharomyces cerevisiae, that is a brewer's yeast which is mainly used in the fermentation process of alcohol, which finally produces ethanol or ethyl alcohol. So that is organic acids and alcohols. Now we will discuss enzymes. Yeah, what are enzymes? You know that enzymes are the proteinaceous substances of biological origin and which have the capacity of catalyzing the biochemical reactions without undergoing any change. You know, whenever a biochemical reaction has to be occurred, that biochemical reactions will be catalyzed in the process of enzymes only. So enzymes are mainly involved in biochemical reactions. That's why these are proteinaceous substances and have the capacity of catalyzing biochemical reaction. And many microbes are involved in the production of many enzymes which are mainly used in the industrial purpose as well as in the medicine. So we'll see which are the enzymes obtained from the microorganisms. The enzymes like pectinases, proteases, lipases and streptokinase. So all these enzymes are very, very important. Pectinases, proteases, lipases and streptokinase. So pectinases. These pectinases are obtained from the fungal cultures of Aspergillus niger. So as I said, Aspergillus niger which produces citric acid, the same Aspergillus niger which produces pectinases. So these pectinases are mainly obtained from the fungal culture. That fungal media actually is called as Martin rose and Bengal agar media. The fungal media is called as Martin rose and Bengal agar media, which is mainly containing a pectin. So in that pectin containing media, this Aspergillus niger produces an enzyme called as pectinase. Then what is the use of pectinase? Why it is used? That pectinase is very, very important. So pectinases are mainly used in the extraction of fruit juices from fruits. In order to extract the fruit juice from fruits, they use pectinase, and even they are used to remove the bitterness from fruit juice. The bitterness in the fruit juices will be removed with the help of pectinases, and these are even used in the fermentation of green coffee. These are certain uses of pectinases. The next one, proteases. These proteases are obtained both from bacteria as well as fungi. The bacteria name is called as Bacillus subtilis, and the fungi name is called as Aspergillus oryzae. Aspergillus oryzae, it is a fungi, it is a bacteria. So proteases can be obtained from both bacteria and fungi. Bacteria is called Bacillus subtilis and fungi is called as Aspergillus oryzae. Then, what are the uses of proteases? These proteases are mainly used to increase the tenderness of meat. See, the meat can be stored for a certain period of time. If they are treated with proteases, they will maintain the tenderness in meat. So they will increase the tenderness of meat. And these are also used in the extraction of fish liver oil. To extract the fish liver oil, they will use proteases. They are also used in alcohols and also used in softening proteases. And you may have noticed that whenever you brought the fruit juices from the market, so the bottled fruit juices are more clearer when compared to the fruit juices you made at home. You just compare. You prepare a fruit juice at home and you bring a bottled fruit juice. When you compare both, the bottled fruit juices are more clear because they are treated with pectinases and proteases. This is very important. So why the bottled fruit juices are clearer? Because they are treated with pectinases and proteases. Next one, lipases. So these lipases are nothing but these are lipid digesting enzymes and are mainly obtained from Candida lipolytica, geotrichum and mucus species. Lipases are mainly obtained from Candida lipolytica, geotrichum and mucus species. Then what is the use of lipase? See, these lipases are used in the flavoring of cheese. To provide flavor to the cheese, they will use lipases. And another important advantage of lipases is that they are mainly used in detergents. What the soap, shelf, etc. you are using at home, in those detergents, they are mainly using these lipases. So what is the advantage in using detergents? So the lipases are mainly involved in removing the oily stains from the laundry. So any oily stains occurred on the cloths that will be removed with the help of detergents, which means lipases are used in detergents and which are mainly able to remove the oily stains from the laundry. That is about lipase. And next one, streptokinase. This is more important among these enzymes. So streptokinase is one of the important enzymes obtained from streptococcus. So streptokinase is the important enzymes obtained from streptococcus, and this streptokinase is also called as a TPA, that is nothing but tissue plasminogen activator. Tissue plasminogen activator. And the main function of streptokinase is that it is used as a clot buster. So this is very important. The main function of streptokinase is that it is used as a clot buster. Do you know the clotting mechanism that you studied in the body fluids and circulation? So usually how the clots will occur? Whenever there is a small cut, initially bleeding will occur, later clots will be formed. So during that coagulation, prothrombin gets converted into thrombin. Later thrombin will convert the fibrinogen into fibrin threads, and fibrin threads along with the blood cells, they will form the clot. So that clot usually occurs when it is exposed to air. But even inside the blood vessels also sometimes the clots will be occurred. They are called as internal clots. The clots which are formed inside the blood vessels are called as internal clots. If for example, if this is the blood vessel, if there is a clot found here, so there is a clot formation occurs inside the blood vessel, what happened? The blood flow will be restricted. If for example, this is a coronary artery or any one of the blood vessel which will carry blood to the heart, oxygenated blood to the heart. If there is a clot occurred, that flow of blood is restricted so that it leads to myocardial infarction or heart attack. So in those patients, the patients suffering from myocardial infarction, they may be having internal clots, and to remove or to dissolve these internal clots, they will give one important enzyme called as streptokinase. So this streptokinase, it will remove or dissolve the internal clots inside the blood vessels. That's why it is called as clot buster. Then why it is called as a tissue plasminogen activator? Plasminogen, it is another plasma protein which has fibrinolytic effect. Fibrinolytic effect means it will dissolve these fibrin threads and remove the clots. That's why it is called tissue plasminogen activator. It is very important. Streptokinase obtained from streptococcus, also called as a tissue plasminogen activator. The main function of streptokinase is that it is used as clot buster to dissolve the internal clots which are present in the blood vessels bioactive molecules. What are bioactive molecules? These are the molecules which are active or functional only in living systems or can interacting with the living components. So we need to discuss two important bioactive molecules. One is called cyclosporine A. Cyclosporine A. The other one is called statins. So these are the two important bioactive molecules. So first one, cyclosporine A. It is one of the important bioactive molecule obtained from the fungus Trichoderma polysporum. So cyclosporine A, it is obtained from the fungus Trichoderma polysporum. Then, what is the use of this cyclosporine A? The cyclosporine A, it has antifungal, anti-inflammatory and immunosuppressive property. Antifungal in the sense they will fight against certain fungi. It is a fungi. It is obtained from this fungi which will be fighting against some other fungi. And anti-inflammatory. So whenever irritation occurs, that is called as inflammation. So they will act as anti-inflammatory and they have immunosuppressive property. Immunosuppressive property in the sense they will suppress the immune system. So when they need to suppress the immune system, so cyclosporine A, it is extensively used as immunosuppressive agent during organ transplantation. So this is very, very important. Immunosuppressive agent. Immunosuppressive agent during organ transplantation. So organ transplantation, for example, you just imagine one person required kidney. So another person will be donating a kidney. So you are transplanting a kidney from one person to another person. So you cannot simply transfer or transplant the kidney from one person to another person. So firstly, we need to check the blood groups. The blood groups has to be matched. Later, various components are also being matched. Even though the blood groups are matched, when we transplant a kidney from one person to another person, the immune system of this person cannot simply accept that organ because whatever the materials which are coming from outside, they will be treated as a foreign particle by our immune system. So whatever the components we have, that will be accepted by our immune system. But when a dust or any other particles will come, that will be treated as a foreign particle. Even the kidney of another person will be treated as a foreign particle by our immune system. So what the immune system will do when you transplant the kidney from one person to this person? So in this person, the immune system will start producing T lymphocytes, and those T lymphocytes will destroy that organ. So in order to avoid such circumstances, the cyclosporine A, it is used as immunosuppressive agent during organ transplantation. So this cyclosporine A, it will inactivate or it will suppress the activation of T lymphocytes so that the organ can be accepted or it will be transplanted. That is one of the major importance of cyclosporine A. You should remember cyclosporine A obtained from the fungus Trichoderma polysporum and it is used as immunosuppressive agent during organ transplantation. And the next one is called statins. Yes, statins are also obtained from the fungus, especially a type of yeast. The statins are obtained from the yeast which is called Monascus purpureus. So statins are mainly obtained from the yeast Monascus purpureus. Then what are the mainly used as a blood cholesterol lowering agent. So these are about the chemicals, enzymes and bioactive molecules. Whatever the components, whatever the things I have discussed now, these are very important for your examination, and even few questions from this topic have been asked in your NEET examination that has been conducted in the last month, sorry, in this month. So this is very important. You should remember and should note down the points which I have covered in this topic. Now let me discuss a few objective questions related to this topic. Yeah, first question. Renin or chymosin obtained from Mucor Mucor myceli and Endotheo parasitica is specifically used for the production of. Have you heard about renin or chymosin? This I have not discussed. Renin or chymosin is another type of enzyme which is mainly extracted from the stomach of calf. So that is mainly used for option A cheese, option B cottage cheese, option C camembert cheese, option D roquefort cheese. You know different types of cheese. So this renin enzyme is extensively used in the production of cottage cheese. When you look at the images, you will come to know. So in the cottage cheese production, they will use chymosin or renin. Next, second question. Probiotics are option A cancer-inducing microbes, option B new kind of food allergens, option C live microbial food supplement, option D safe antibiotics. Here probiotics are nothing but they are the live microbial food supplement. Sometimes the live microbes will be used as a food supplement. They are called as a probiotics. You will study about these probiotics in the biotechnology chapter. Next, third question. Which cheese is commonly referred to as hard cheese? Option A cheddar cheese, option B roquefort cheese, option C camembert cheese, option D cottage cheese. While explaining about the cheese, I told you soft cheese, semi-hard cheese and hard cheese. Here hard cheese is called as cheddar cheese. So this cheddar cheese is a hard cheese. So this can be cut into pieces. Next, fourth one. Idli is a fermented product of. So idli is fermented product of option A rice, option B maize, option C barley, option D black gram. So here rice, black gram both are correct, but so only with the help of rice that fermenting activity will not be induced. With the help of black gram, that black gram will induce that fermentation activity. That's why here correct action will be black gram. And fifth question, the commercial production of ethanol is carried out by. During the commercial production of ethanol, what the organism will be used? Option A Fusarium, option B Candida albicans, option C Saccharomyces cerevisiae, option D Candida utilis. You know that Saccharomyces which is commonly used in the production of ethanol. So these are few objectives. Remaining objectives I will send in the group. So that completes today's class. And next class is also very important. We will discuss in the next class.